Assalamualaikum and hi, this is BSM 4301 Bioinformatics. So today I'm going to present to you practical 5. We have practical 5.1 multiple sequence alignment using bioedit interface, practical 5.2 cluster omega multiple sequence alignment, practical 5.3 praline multiple sequence alignment, and practical 5.4 espresso at tea coffee server. So we are from group 7. There are 3 group members of this group.
Assalamualaikum and hi. I am the leader for practical 5. My name is Sofia binti Zulkifli and my matrix number is 199172.
My name is Lo Shenzi. My matrix number is 197317.
Hi and Assalamualaikum. My name is Mama Faiza bin Zakaria and my matrix number is 19662. Bye!
So we're going to start with practical 5.1 multiple sequence alignment using bioedit interface. Firstly, you need to browse the URL address of NCBI on the World Wide Web. Next, you need to retrieve the amino acid sequence using gene bank accession number of AY260764. Insert it into this column, and when the output shows, you need to scroll down to find the amino acid sequence here. Next, you need to browse for signal P4.0 server to locate the signal peptide. And you need to insert the amino acid sequence of AY260764 that we have been retrieved before into this column. And this is the output of signal P4.0 server. It will show you a graph. When you scroll down, it will state the measure, position, value, cutoff. Signal peptide, yes. Cleavage site between position 18 and 19. This is the cleavage site on the graph. And there are 2 hashtags below, which are process FASTA entries and GFF file or process entries. You need to click on the process FASTA entries and you will retrieve the amino acid sequence of mature lipase. And you need to browse the URL address of BLAST. Make sure it is BLAST-TP. Click on the BLAST-TP and paste the amino acid sequence for mature lipase that we have been retrieved from signal P output into the BLAST-TP window. Click on BLAST and the result will showing. It will show you 100 sequences. However, we need to change the algorithm parameters. The maximum target sequences change to 5000 and scoring metric used is Blossom 45. This is due to produce sequences with different percentage identities, and it will produce 5000 sequences. Click on certain sequences needed and click on the alignments. Due to only 2 sequences needed from every percentage identities, so after that we need to click on gene bank, click on FASTA, and we'll retrieve the amino acid sequence of desired lipase. So we need to copy all of the sequences needed into one document. However, we also need to save the FASTA by creating the file. Beside the amino acid sequence of desired lipase, we can see the send to. Click on the send to, click on file, click on create file. So we can save all of the sequences in one file. Next, we need to deposit all the sequences into BioEdit. You need to download the BioEdit app into your laptop. Select all sequences and perform multiple sequence alignment. The selected sequences will be highlighted in black color. To perform multiple sequence alignment, we need to click on the accessory application and click on the cluster W multiple alignment. And check the box for output cluster format with cluster consensus sequence generation. This is the box, and click on the run cluster W, click on OK, and the result will be shown. And we need to analyze the alignment. This is the enlargement of the alignment. Then we need to annotate the functional motif based on the information obtained from this article. You need to search for this article. So this is the result or output of finding functional motif. We have zinc binding site, catalytic triad, calcium binding site. So we move on to practical 5.2, cluster omega multiple sequence alignment. Firstly, you also need to browse the URL address of cluster omega and paste the amino acid sequences in FASTA format into this window. Insert the query sequence. Click on the submit button, and you also need to download the alignment file. Click on the download alignment file, and this is the downloaded alignment file, and it also showed the output of finding functional motif. We also need to click on the result summary to display the result files. It will state the input sequences, tool output, alignment in cluster format with base or residue numbering, guide tree, phylogenetic tree, and percent identity matrix. And we need to click jar view to display the alignment in color format. Click on file, click on input alignment, click on from file. Retrieve from the file that we have been saved all 10 sequences and plus the query sequence. So this is the output of multiple sequence alignment with jar view in a color format. So we move to practical 5.3, Praline multiple sequence alignment. You need to browse the URL address of Praline. This is the page. And you need to paste all of the sequences obtained from practical 5.1 into the window here. Also include the query sequence of the lipase. Click on the submit button. Below that we have Praline run. Click on the Praline run. So the results will be displayed in different formats. There are 4 formats: residue type, conservation, hydrophobicity, secondary structure. So this is the results color-coded according to the cluster at residue color scheme. And this is the results color-coded for amino acid conservation, results color-coded for secondary structure, and the last one is the results color-coded for hydrophobicity. So we move on to the last practical 5.4, Espresso at Tea Coffee Server. You need to browse the URL address of Tea Coffee Server. As we know, Espresso incorporates all the available structural information in your alignment. It will process the best sequence alignments if the structures are available. So you need to click on the Espresso button here, and this page will come out. This is the column where you need to input your sequence to align. Paste all of the sequences obtained from practical 5.1 into the window, all of the 10 sequences that we have retrieved, and also the query sequence. Scroll down, and you need to make sure you tick on the automatically fetch PDB templates here, and click on the submit button, and this is the output that will be shown out. However, you also need to check the cluster alignment and template list files. Scroll down after you retrieve the output, and you can see the cluster W alignment file and template list file. Click on both of them, and this is the results after click the cluster alignment, the output of multiple sequence alignment, and also the functional motif. This is the template list file. Note that the template list file is a program managed to associate with your original sequences or your query sequence. If this file is empty, it means no structure was available to align your sequences. However, when we click on this template list file, this file is not empty, so we have the structure that available to align our sequences. Thank you. That's all from me. Thank you.